Hey everyone, welcome back to the Deep Dive. We love to get into the nitty-gritty here, and today we're going deep on rational drug design.
Yeah, sometimes called structure-based drug design, or SBDD for short.
Right, SBDD. So we all want new medicines, better treatments, but the old way of finding them, kind of like throwing darts in the dark, right?
It was very much a trial and error process. You'd screen thousands, even millions of compounds, hoping to get lucky.
Huge libraries of chemicals, hoping one sticks. So how is this rational design different?
It's much more targeted. Imagine you're trying to design a key for a lock. The old way would be to try every key you could find.
Ugh, frustrating.
Right. But if you could actually see the lock, the shape of the keyhole, you could design a key that fits perfectly.
Makes sense. So with rational drug design, we're looking at the actual structure of the disease target, right? Usually a protein.
Exactly. We're figuring out its three-dimensional shape, understanding its vulnerabilities. Then we can design a drug molecule, that key, to fit snugly into that target and disrupt its function.
So we need to identify that target first, right? Like, what's the bad guy we're going after?
Absolutely. Target identification is the crucial first step. Scientists look for proteins that play a key role in the disease process. You want something that, if you could hit it with a drug, would really make a difference.
Like a protein that's essential for a virus to replicate or something.
Exactly. Or maybe a protein that's driving uncontrolled cell growth in cancer. Then there's this concept of drugability.
Drugability, what's that?
It means, can we actually design a drug against this target? Does the protein have a pocket or a groove where a small molecule drug can bind?
Okay, so we've got our target. We know it's druggable. Now, how do we actually see this protein? These things are tiny.
Right, we're talking about molecules here. For a long time, X-ray crystallography was the gold standard.
I've heard of that. You have to grow crystals of the protein.
Yeah, which can be really challenging. But if you can get good crystals, X-ray crystallography gives you a really detailed picture of the protein's structure. But there are other techniques too.
Like what?
NMR spectroscopy is great for smaller proteins. It can tell you about how the protein moves and changes shape in solution. Then there's cryo-electron microscopy, or cryo-EM.
Cryo-EM, that's a newer technique.
It is, and it's really revolutionized structural biology. It's especially good for large, complex proteins, even membrane proteins, which are notoriously difficult to study.
I've also heard a lot about AI being used in this field.
Oh, absolutely. Artificial intelligence is having a huge impact. Tools like AlphaFold and RosettaFold can now predict protein structures with incredible accuracy, even for proteins that haven't been experimentally determined.
Wow. So AI is like a shortcut.
In a way, yes. It can give us a starting point for drug design, even if we don't have a perfect experimental structure.
So we've got our target, we've got its structure. Now we can actually start designing the drug.
Right. This is where computational modeling comes in. We use sophisticated software to see how different molecules might fit into that target binding site.
So are we just like throwing a bunch of molecules at it virtually?
Not quite. There are a few key techniques. Molecular docking is a big one. It's like a virtual screening process. You take a library of compounds, maybe thousands or even millions, and you dock them into the target protein structure on the computer.
I see. So the computer is predicting how well each molecule will bind, right?
Exactly. And how they might interact with the protein at the atomic level. Then you can rank these compounds, prioritize the ones that look most promising.
What other tools are in the computational toolbox?
Well, molecular dynamics simulations are really important. These let us simulate how the drug and the target protein move and interact over time. It's not just a static picture anymore. We can see how things wiggle and jiggle and how that might affect binding.
That's cool. So it's getting even closer to what's really happening in the body.
Yes. And there are even more advanced computational methods like free energy calculations that can give us even more accurate predictions of binding strength.
Okay, so we've used our computational tools. We found some molecules that bind really well to our target. Are we done? Is that our drug?
Not quite. These are just starting points. We call them lead compounds.
Lead compounds. So what happens next?
Lead optimization. We need to make those lead compounds even better. Maybe we need to make them more potent, meaning they bind even more tightly to the target.
Right. So a lower dose is needed for it to be effective.
Exactly. Or maybe we need to make them more selective, meaning they only bind to our target and not to other proteins in the body. That helps reduce side effects.
Makes sense. Anything else?
We also need to think about how the drug will behave in the body. How well is it absorbed? How is it distributed? How quickly is it broken down?
Right. All those important things.
Yeah, it's all about finding the right balance of properties.
And how do we optimize the lead compounds? Is it all done on the computer?
Computational tools are still really important here. We can use them to predict how changes to the molecule structure will affect its binding affinity, its selectivity, its ADME properties, but it's also a lot of lab work.
Like what?
Chemists will actually synthesize those modified compounds. Then we test them in the lab, in cells, sometimes even in animals to see how they behave. We also often obtain crystal structures of the optimized drug bound to the target.
To see if it's really binding the way we predicted?
Exactly. That's a key part of structure validation. We want to make sure our computer models are accurate.
So it's this constant interplay between the computational world and the real world, the experiments.
That's right. It's an iterative process. Design, test, refine.
Cool. So could you give me some examples of drugs that have been successfully developed using this rational design approach?
Absolutely. One of the early success stories was HIV protease inhibitors. These drugs were designed to target a key enzyme that HIV needs to replicate.
And they were a game changer for HIV treatment, right?
They were. Another great example is Tamiflu and Relenza.
For the flu.
Right. These drugs target an enzyme called neuraminidase, which is essential for the flu virus to spread.
So they're designed to stop the virus from getting out of infected cells and infecting other cells.
Exactly. And then there's Gleevec, or imatinib. This drug was a breakthrough for treating chronic myelogenous leukemia, or CML.
I've heard of that. It targets a specific protein that drives the uncontrolled growth of white blood cells.
Yes. Gleevec was one of the first examples of a targeted cancer therapy, and it's had a huge impact on CML treatment. And there are many more examples. Venetoclax, for example, is a drug that targets a protein involved in apoptosis, which is programmed cell death. It's used to treat certain types of leukemia and lymphoma.
So these are all really significant drugs. What would you say are the main advantages of this rational drug design approach?
Well, for one, it's much more efficient than the old way of just randomly screening compounds. We're not just hoping to get lucky anymore. We're using our knowledge of the target to guide our search.
So less time, less money wasted.
Potentially, yes. It also allows us to go after targets that were once considered undruggable.
So proteins that just didn't have a good binding site for a small molecule drug.
Right. But with rational design, we can sometimes find ways to target those proteins, maybe by designing a drug that binds to a different part of the protein or that disrupts protein interactions.
Cool. Any other advantages?
Well, I think it also gives us a much deeper understanding of how drugs work. We're not just throwing a chemical at a cell and seeing what happens. We're actually seeing how the drug interacts with its target at the molecular level.
That's got to be helpful for designing even better drugs in the future.
It definitely is.
Okay, so it sounds like rational drug design has a lot going for it. But there must be some challenges, right?
Oh, of course. It's not a magic bullet. One challenge is that getting good quality structures of the target protein can still be difficult.
Like growing those crystals for X-ray crystallography can be tricky.
Yeah. And sometimes the protein might be really flexible, and it's hard to capture that flexibility in a static structure.
I see. Like it might change shape when it binds to the drug.
Right. Exactly. And our computational models are not perfect. They're getting better all the time, but they can still make mistakes.
I guess it's hard to simulate everything that's happening in a living cell.
It is. And then there's the whole issue of ADME absorption, distribution, metabolism, excretion.
Right. We talked about that before.
Predicting how a drug will behave in the body is still a major challenge. It's not just about binding to the target. We need to make sure the drug gets to the right place in the body, that it's not broken down too quickly, that it's eliminated safely.
It's complex.
Very complex. And of course, some targets remain truly undruggable, even with the best rational design techniques.
So there's still work to be done. What do you think the future holds for rational drug design?
Oh, I think it's incredibly exciting. We're seeing so many advances. Artificial intelligence and machine learning are really transforming the field. They're making our computational models more powerful, more accurate.
So could we get to a point where we don't even need to do experiments anymore? Everything's done on the computer.
I don't think so. I think experiments will always be important. But AI can definitely accelerate the process, help us prioritize the most promising drug candidates.
Cool. What else is on the horizon?
Well, we're also seeing a trend towards integrating more and more data into drug design, not just the structure of the target protein, but also genetic information, clinical data, even patient-specific information.
So tailoring drugs to an individual's unique genetic makeup.
That's the goal. Personalized medicine, it's still early days, but I think it has huge potential.
Yeah, that would be amazing. So rational drug design, it's more efficient. It's allowing us to go after tougher targets. It's giving us a deeper understanding of how drugs work. And with all these new advances like AI and personalized medicine, it really seems like the future of drug discovery is bright.
I agree. It's a really exciting time to be in this field.
That's a great thought to end on. Thanks so much for taking us on this deep dive. With all the innovation happening, you have to wonder how much faster could we be discovering new life-saving medicines, say, a decade from now? Something to think about.
It definitely is.
See you next time.